All right. So we are uh, in the digital platform uh, of the of the area of the digital hub. Then as we can see here, yeah, this is the trend research in biodiversity. There are three key points, yeah, nowadays, which is biodiversity, biotechnology because we're using DNA, yeah, and bioinformatics, yeah, how to handle the large data that we generated from DNA. Yeah. So these three key points, biodiversity, biotechnology, and bioinformatics, will be together uh, bring up, yeah, in the future. And the other thing is the biology research is shifting from observation into the prediction. So this is the reason why we need bioinformatics. In terms of data, we work from qualitative into the quantitative. Yeah. So this is the reason why we use uh, DNA and other genomic data. As we can see here that we are in the genomic area. Yeah. Maybe back to uh, starting from 1990, yeah, we are in the genomic area, and then we move to transcriptomic where we use RNA, and then we use uh, for amino acid data, yeah, and then nowadays, yeah, probably in the future we use metabolomic data. So especially for the biodiversity research, we focus use only genomic data. Yeah, why? Because DNA is very stable. Yeah, yeah, compared to RNA and protein. Yeah, which is unstable. So because DNA is stable, we can transfer. DNA is transferable. Yeah, we can transfer from other species without border, without limitation. Animal into bacteria, bacteria into animal, vice versa. Yeah, plant into animal into the plant. Transferable. Yeah, maybe we can actually particularly we can say horizontal transfer because stable DNA can travelable. Yeah, we can bring DNA isolation from the jungle into the lab. Yeah, and then yeah, because they are DNA is very stable and easy to handle. Yeah, without special treatment. When we working with RNA or protein, we need a special treatment, which is very high, yeah, very expensive and recordable. Yeah, because DNA like the tape. Yeah, we can see past living in the past. Yeah, living in how to say the 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 happening in the past we can see from DNA. Yeah, and sustainable. We talk about sustainable because we can isolate DNA from environmental. Yeah, so we can isolate DNA from water, from the soil, and other habitat. So where we cannot easily isolate from the living thing. For example, when we working with lion, how to cap how to capture the lion or tiger, yeah, to take the DNA. Yeah, maybe maybe first of time we must kill kill the tiger, and then we take the blood and then go to bring to the lab and isolate DNA. But because DNA is very sustainable, we don't need to kill the animal. Yeah, we can isolate DNA from their feces, their habitat. This is what we call it as eDNA, environmental DNA. And in terms of functional, DNA is basic unit of information, yeah? Blueprint of the organism. And the other is the DNA provide the hue characters. Yeah, in morphological character, we at least we can get 100, but DNA provide more than that. Yeah, maybe we can get 10,000. Yeah, even 2,000 or more the character. And compared to the morphology, DNA provides best homology. Yeah, we when we doing phylogenetic evolution, systematic, we need homology character, and DNA provided. And of course, DNA will produce more natural differences than other characters. So this is the reason why we use DNA. Okay, there are three genome, yeah, for of the plant, because I working with plant. The genome from nuclear, genome from mitochondria, and genome from plasmid. And we call this discipline, specific discipline, what we call it as a bioinformatics, that we learn about biodiversity, and at the same time we also learn about the evolutionary history. Yeah, so I little bit review about what is bioinformatics. Bioinformatics, there are two approaches in bioinformatics: phenotypic and cladistic. But we focus on cladistic. In cladistic analysis or phylogenetic analysis, we sometimes use the molecule character and non-molecule character. Then we construct phylogenetic tree. Yeah. Then I skip this one. Then we use, because we use very huge characters. Yeah, we need software, yeah, to facilitate our analysis. And then at the end, we will have a phylogenetic tree. In the next several slides, I'm going to show you my experience during my research with the team, of course, how the biodiversity, molecular biodiversity can manage the biodiversity that we have. Taxonomy, in the taxonomy, using the DNA, we can easily to identify, to know the position, the status of the new species, new subspecies, or new cultivar. Yeah, this is our publication this year about apple cucumber. Apple cucumber is actually originated from Karawang, Jember, and Aceh. Yeah, maybe maybe in Padang already heard about apple cucumber, timun apple. Yeah, through the study, by the study using DNA, we definite, we produce the scientific name of the apple cucumber, which is they belong to the Cucumis melon, and consider to be a new subspecies. Yeah. About the apple cucumber, timun apple, the shape, the appearance like apple, but the taste is like a melon. Yeah. Okay, the other research that I and the team conducted is bibliography of the Australia orchid. As we see here in the phylogenetic tree, we can see the Australia here, so we can recognize, oh, this is from Asia, this is from Indonesia, this is from Australia. Yeah. The other thing is still bibliography. We also conducted a bibliography study of Mangifera species. Yeah, as we can see here, this is the Mangifera species from Thailand. The rest of is from Indonesia. So we can keep our Mangifera species. Yeah. The other topic that we conducted is the phylogenetic diversity. Yeah. We can actually setting the priority of conservation. Yeah. If we calculate, if we calculate the branch, the red or the branch, which color, with red branch, we can see here this is very high phylogenetic diversity, which is uh, which is the this is very variation. Yeah, very many variation, and we don't need to conserve this one. But the below one, very low phylogenetic diversity. So we have to conserve this species that we have to conserve. Yeah. Another thing is genetic improvement using the biodiversity study. So in our research and published in 2008, we already generated a new clade of irides. So we can cross each other. Yeah, for example, because irides and panda is belong to the similar clade, we can cross each other. Irides and panda, we produce the name hybrid aeropanda. And for example, there are four genera, which is panda, rhinconceros, neophretina, syndrome, cross each other, produce darwinara. So this is how to, yeah, this is in horticulture program, yeah, to 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 improve the breeding. Okay. The other is the topic about phylogenetic screening to find out the new drug. For example, there is one species which is limited distribution only in South Asia, in India, Bangladesh, and Sri Lanka. Yeah, and we have to find out the relative from Indonesia. So we don't need to go to India, Bangladesh, and Sri Lanka to take the tree. That's why ashwagandha is important to cure the cancer. So using the phylogenetic tree, we can see the relative of Titania somnifera, which is ciplukan, ya, atau cecendet, ya. Di Sunda, di Jawa, we call it as cecendet, but in Indonesia we call it as ciplukan. So ciplukan is relative with Titania. So we can conclude that physalis or ciplukan can be used to cure the cancer. Yeah, they have potential to in cancer. DNA barcode. Yeah. As we can see here, there are several species that I identified as the box. Yeah, a different color in the box. 1, 2, 3, 4, and we can develop the primer to identify this kind of species. For example, at the end of the process, we can design the primer to identify the species. For example, number 1 here, yeah, black, yeah, with black color. They have this band around 190 base pair, but the other box will have about 2020, and the last one, 4, in the species 4, we can see here under 100 base pair. This is the barcode. Yeah. Thank you. I hope you enjoy my presentation. I
Thank
will back
you.
to the Fitra.
Yeah, thank
Assalamualaikum
you.
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for interesting presentation from Professor Topik. Now we open the discussion. Maybe we have about 1 minute because based on the schedule, we will close this invited speaker session to 2 p.m., 2 o'clock p.m. So if you have question, please write it in the chat box, or you can ask the question directly to Professor Topik.
Yeah, because only 1 minute, yeah, Pak Fitra.
Yeah.
Not enough time. Not enough time to
Yeah.
to question and to answer, of course.
Yeah. While waiting for the question, maybe I want to ask one question for you about the apple cucumber.
Yeah.
This is interesting because the cucumber, but the taste is like a melon. But it is considerably become a subspecies. Yeah. Why don't you put it into new species? Why it should be in subspecies? Is there any threshold score for determining a group of species become subspecies or become a new species?
Okay, thank you, Fitra, because so many consideration, yeah, that we have to take before we decide this is the new species. This is not new. Why we decide this is the subspecies? Because they can breeding each other with the melon. But if we decide if we put in new species, they cannot breeding each other. That's very simple answer.
Okay, okay, I understand.
Okay. Yeah. Yeah. If subspecies, they can still able to cross. But if we new species, of course, apple cucumber cannot crossing over with melon itself. So that is the answer. The other thing is we need to consider the stability. That is the stability of the offspring. Yeah, especially in F1, in F2, F3, F3, because it's still under study. Yeah, so we do not put them in the varieties, new varieties. Yeah, because they do not show the stability in the culture, in the field. So we put there in I think it is very fair, yeah, very fair for apple cucumber in subspecies. That's all.
Okay, thank you, Professor Topik. And I think that's all the question, because the schedule is very tight. So I have to close this session, because we have 2 more sessions from the participants. And thank you very much once again to Professor Topik for coming to this Zoom meeting. And you may leave the Zoom if you have another agenda today. And thank you very much once again.
Okay, thank you.
Yeah, thank you, Professor.
Wassalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, ladies and gentlemen, in the chat box, you can see the link for the attendance list. You can tick your presence by clicking the link. So your presence will be recorded in this Zoom meeting. Don't forget to tick your presence. And now we will see the recorded video presentation from all of you.
is Dr. Adibah binti Abu Bakar. Assalamualaikum and good day to everyone. My name is Dr. Adibah binti Abu Bakar, and my research today is about development of species-specific chinchilla species EDA primers for rapid alien invasive species AIS monitoring. I would like to start off with the highlights. What is actually invasive alien species? So invasive alien species are plants, animals, pathogens, and other organisms that are actually non-native to an ecosystem. However, it will cause economic or environmental harm or adversely affect human health. Particularly, it is actually have an impact to our biodiversity and are making decline to our native species. So the major reason for the introduction of the alien species in Malaysia is actually due to recreational fishing. The most prominent example is the peacock bass or the species named chinchilla. It is intentionally released into rivers or lakes by former mining areas or by irresponsible anglers. And because peacock bass is actually a good sport fish, however, their uncontrolled spread and unintentional release cause negative impact globally, especially to Malaysian native fish species. So the peacock bass in Malaysia is labeled as alien invasive species or AIS. It is an urgent need to monitor AIS to preserve our native fish species. But currently, local authorities tender to common method of long-term surveys and large fishing catchment. This is called visual field capture techniques and is often to be considered costly and labor-intensive. So in my experiment, in my research, I'll do 3 key indicators whereby the first thing our team will identify the species level. This is to make sure that we clear any ambiguities by the taxonomy results, and this will help to correctly identify the species that we are going to research. Next, we develop a species-specific primer using cytochrome oxidase 1 gene. So this will be a diagnostic segment to identify the species only by using fragmented environmental DNA, or we call it here eDNA. Lastly, we will do the primer validation. We will do in situ analysis through primer blasts and age analysis, and we are looking forward to make sure that our primers do not have any cross amplification to other species. This is an example of our workflow. We start off with the environmental sample. We collect it through water, and then we'll do DNA extraction and PCR. After that, we will barcode the sample and we will prepare primer and probe to detect 2 target species. We use 3 types of sequences we found from the gene bank, and the details are as in this slide. As a result, we actually developed 18 primers comprising of different species-specific primers for the 3 species that we are looking to identify. Okay, the first one is for Chinchilla cavalry, CK. The next one is Chinchilla monoculus, CM. And the third one is Chinchilla oscillaris, CO. We use primer blast tools to design our primer, and we have adjusted the parameters to 5 compulsory criteria. The first one is our primer must contain 18 to 23 nucleotides. Second, the amplicon must be between 280 to 400 base pair. The third one, it must have a minimum of 2 mismatches only at the 3 end. and the primer melting and aniline temperature must be between 50% to 60%, and lastly, the GC content must be between 20% to 60%. We have run our developed primer in two sites. Field site number 1 in mini Amazon Kampong Bang Perak, and in the field site number 2 in Tasik Rabab. We successfully identified Chinchilla auxiliaries and Chinchilla cavalry by only using our primer. Therefore, without catching these species physically, we actually can detect the presence of the species by only using environmental DNA. Again, we have isolated the eDNA from water sample, and we found out that the longest period that a certain eDNA can be kept to have a good amplification is actually between 1 to 22 days. After that, the intensity of the eDNA will decrease. So we suggest for our audience, if you are going to use eDNA, make sure the storage is between, if you want to extract it, it is between 1 to 22 days. If you extract it after, then you will get less amplification. We also have validated the primer and probe that we've designed, and Alhamdulillah, we found that for the least a minimum detection of 0.625 nanogram sample can be detected for identification of the species in the environmental DNA. So we would like to confirm here that our species-specific marker do not amplify non-target species. There is no cross-species amplification, if you can see from here. And this proof we suggest to use eDNA to detect presence of invasive species because it is environmentally friendly. For example, we only collect standard volume of water, and then it is very economical and less time consuming. You do not need long-term survey sampling. Next, it is non-invasive because you do not need complex tracking equipment and you do not damage the environment. So the way forward, what we expect from our product is that it can be used to address standard management control as above. This data is collected from Department of Fishery. So by collecting the data that we have now, we are looking forward to inform in Malaysia that the peacock bass must be managed properly so that it will not become invasive towards the native species. If you have further inquiries and for further collaboration, you can contact me at atiba@fsmt.upsi.edu.my at the Genomic Lab, and we are very thankful to the Department of Biology, Faculty Science and Mathematics in UPSI and our FRGS grant for the continued support to our efforts to contribute to the SDG. Thank you very much.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity is given to us. We are presents our study about RBCL chloroplast genes primer designs as DNA barcode for Ipomoea batatas as alternative food for people with diabetes mellitus. The members of the study are Samsu Rizal, Afifatul Akhir, and Elsa Badria. We are from Biology FMIPA Universitas Negeri Padang and Fakultas Farmasi Universitas Andalas. The background of Ipomoea batatas has high productivity, 25 tons per hectare, wide adaptability to various environmental stress, resistant to pests and disease. The harvest period is short, which is between 3 until 4 months. Sweet and sour tuber texture. Tubers have low crude fiber content. Tubers have a high nutritional content. Plain character according to industrial needs. Source of carbohydrates, high calories. The fourth carbohydrate content after rice, corn, and purple sweet potatoes. Source of vitamins and minerals. Vitamins consist of A, C, B1, and B2. Minerals consist of ferrum, phosphorus, calcium, and sodium. Protein, fat, crude fiber, calories, and carbon. Taxonomy of Ipomoea batatas, Kingdom Plantae, Divisi Magnoliophyta, Class Magnoliopsida, Order Solanales, Family Convolvulaceae, Genus Ipomoea, Species Ipomoea batatas. This is barcode regions of plant. Condition of DNA barcoding has to discrimination, universality, and robustness. Based on the recommendation of Consortium for the Barcode of Life, BOLT, in Plant Working Group, the unique DNA barcode for plant identification is the chloroplast MET-K, Materaceae K, and RBCL genes, or ribulose 1,5-biphosphate carboxylate, or ribulose 1,5-biphosphate carboxylate chloroplast. The methodology are first collecting samples. Fresh young leaves, apical leaves, or leaves at the three uppermost nodes, wrapped in aluminum foil and kept in an ice box, preserved in 4 degrees Celsius for 1 week. Second, DNA extraction, DNEZ plant medikit, Chiagen or simplified CTAB or SDS protocol, based on Kitan Tiyawati et al. 2014. Third, DNA barcoding PCR and electrophoresis, chloroplast genome RBCL, based on Watu et al. 2016. Sequencing, Macrogen Korea and first base Singapore or Malaysia. Bioinformatic analysis is BLAST, BLAST, and phylogenetic tree using Genius software. And last is submit sequence to NCBI to get accession number for each sample. And this is the result of design primer produced 816 BP. This is table for quantity and quality of sweet potatoes DNA extracts. We choose for 5 samples. And this is the result of electrophoresis, electrophoregram of RBCL PCR products. Level 1 is madu pucuk hitam, second hitam pucuk, third madu Papua, fourth 100 hari, fifth white sweet potato, and L 100 pp, DNA leather 100 pp. This is the result sequences result of RBCL genes. And this is proof that sample switch with the NCBI database. This is a phylogenetic tree. And conclusion, the Ipomoea batatas DNA barcode was made by designing a primer for RBCL. Primer successfully produced a single DNA band across various applicant sites on multiple samples, variations within a species. That's all. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for coming to this presentation, where I will present the result of our research on primer design and PCR optimization for Salmonella sp detection on refilled drinking water sample. Firstly, I would like to explain about introduction. This research is motivated by the importance of the need for drinking water for the human body. Drinking water must be safe for consumption by taking into account the physical quality, chemical, and bacteriological. And because of the increasing need for drinking water is the safe, suitable consumption in the community, people use drinking water that comes from the amino. The amino is used an alternative because then you must also pay attention to the quality recruitment and standard of drinking water by carrying out water treatment, including filtering, disinfection, and filling process. This done to comply with the regulation of the Keputusan Menteri Kesehatan 907 of 2002 that one of the primary of drinking water suitable for consumption is not polluted by Salmonella sp. And test for the release of pathogenic microbe in water are generally carried out is using microbiological method as most probable number. However, for testing with this method is has a weakness. Therefore, tests to detect pathogenic microbe carried out in this study were carried out molecularly using the PCR technique. In order for the PCR technique to be optimal for detecting coliform bacteria such as Salmonella sp, then one of the important things to do is the design a primer. The primer design is done because many publications of primer design for commercial circulation are still not optimal, such as primer have no specificity and can form secondary structure, such as dimmer, and can only hybridize in a limited temperature range. Therefore, a primer design was carried out to ensure this performance by performing optimal validation experiment. This can save time, money, and help avoid process failure or inconsistent experimental data. Secondly, this material and method in the research. In research material is a complete genome sequence of Salmonella sp, genus prime bioinformatics software, and use thermocycle engine for PCR process. Therefore, the method is the result of firstly primer design with the genus prime software. Secondly, primer is designed with the info target gen. Thirdly, the primer is designed based on ideal primer criteria. Then the primer the design will be tested for specificity and in silico PCR. And lastly, PCR optimization was carried by performing annealing temperature gradient. Thirdly, a result and discussion, the complete genome sequence of Salmonella sp and sequence length of 26.5 base with the info target gen found on the NCBI pack design, and the result obtained were the length of the primer select was 20 and 23 base. The difference between forward and reverse primer temperature rating is not more than 50. It is only 0.2. So it good for the amplification process. And base content of G and C forward is 47.8% and reverse is 55.0%. This, of course, has met the ideal primer criteria. The primer design in the study considered the umbilical structure and ensured the primer target set were from secondary structure. Okay, then the specificity of the forward primer and reverse primer was reached with the primer blast on the NCBI pack. From the table 2, it appears that the primer will only amplify of Salmonella sp and Salmonella enterica. Design primer cannot amplification of 30% of bacteria template based on the result for testing at NCBI. Next, the result of the in silico PCR software, the primer attack to 150 to 110 nucleotide for Salmonella forward primer and 797 to 816 nucleotide for Salmonella reverse primer. This primer will be amplified the invagen region located on chromosome DNA, so 559 base pair. These results are consistent with the prediction of PCR product size with blast primer. The result of PCR optimization, the annealing temperature gradient used to 50 to 60. The result of PCR product for Salmonella sp and then temperature gradient showed positive application result. This indicates that the primer can either well to the target template using a test annealing temperature. Then the annealing temperature of 99.5 Celsius. Number A is used because it has a band size according to the target amplicon, what is more or less 559. compared to the outer and is classified as high temperature high annealing so that it will amplify a specific target out of the band thickness is thinner than outer temperature. Conversion, this research has succeeded in design specific primer for salmon watt and reverse which produce 559 base per amplicum and PCR optimization result obtained the optimum temperature is 59.5 Celsius. Finally, author expert has high appreciation and gratitude to the research and community service Institute of Universitas Negeri Padang. And thank you to Dr. Yuni Ada, SSCMSC, and Dr. Dwi Hilda Putri, SSCM Biomed. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity that is given to extract to the level of mouse liver GSH. The member of this study are me as the lead of the team, Yuni Ahda, and then Siska Alicia Farma, Fadila Humaira, and John Effendi. We are from Biology Department, Faculty of Mathematics and Natural Sciences, Universitas Negeri Padang. Many studies show that Cinnamomum zeylanicum contain antioxidant. Antioxidant is a significant contributor component to our overall health. Antioxidant enzymes have an important role in balancing reactive oxygen species or ROS in the blood. And glutathione or GSH is one of antioxidant enzymes in the body. So the aims of the study is to examine the effect of Cinnamomum zeylanicum crude extract to the level of glutathione or GSH in mouse liver musculus. This is an experimental study. We use 30 mice musculus that were divided into 3 groups. They are control groups, the treatment 1 groups that were treated with 75 milligram per body weight, and treatment 2 that were treated with 150 milligram per body weight of Cinnamomum zeylanicum to the mice for 4 weeks. At the last day, samples were killed by neck dislocation technique. And as long as the treatment, all the samples are fed at libitum. To measure the level of GSH, we use GSH kits. The data then analyzed by one-way ANOVA test. This is the results of our study, the level of GSH. Here you can see there are 3 bars that show the 3 groups of the study. They are control groups, treatment 1 groups, and treatment 2 groups. So the means of GSH level for the control groups are 72.385, and for the means of GSH level for the treatment 1 is 93.872, and the mean of GSH level of the treatment 2 is 106.586. And from the analysis of ANOVA one way, we get that there is the significant difference between GSH level of treatment 2 groups and the control groups. However, there is no significant difference of the GSH level of the treatment 1 groups and the control groups. So the conclusion of this study is cinnamomum zelanicum significantly increased the GSH level in mus musculus liver. Okay, that's all that I could tell you. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Maaf sebentar, Pak. Untuk share screen-nya ada mengalami kendala.
Okay. So research and title assessing underlying diversity using leaf litter plot in a tourism object in Sumatera. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Here I am Fitraria Nugraha with my colleagues Ahmad Muhammad Kadafi and Rujal Satria. We conducted research entitled Assessing Underlying Diversity using leaf litter plot in a tourism object in Sumatera Barat, Indonesia. Papers concerned with the herpetofauna of Sumatera Barat have been primarily taxonomic or inventory. Example, Taney et al. 2010, who work on the inventory of herpetofauna along Sumatra, including Sumatera Barat, and also was et al. 2017, who work on the recuperate frog Pilotus in Mount Marapi and Mount Singgala, and also Arifin et al. Who work on the Pulcrana fantastica. Those study focus on the taxonomic issues and also inventories, and relatively few studies have been focused on the examination of efficient method to obtain particular group of animals like leaf litter frog. This is very important because we have to make the efficient survey to obtain a sufficient number of frog or sufficient number of species to understand the dynamic of the population. We conducted research in Anai Resort, precisely in crown campaign, in the elevation about 612 meters above sea level. Plots were placed on the first floor, spaced by 5 meters each other. Number of plots 45. Size of plot 4 times 4, so it is 16 meters plot, and we survey in each plot for about 5 to 8 minutes by 2 persons. The leaf litter, stones, branches, and wood were overturned to obtain the target species. The total recorded species is 11, representing 5 families, and the total individual number is 20. The richest family is Microhelidae. The highest individual number is Bijurana microbariensis. The density is 2.8 per 100 meter, and the diversity xenon winner index is 2.2. Each plot was completed within 5 to 8 minutes, and no additional new species list after plot 20. And if we look at the other studies using the plot, we can compare this. I can compare this study with the other study. Like, for example, the individual number is higher than Fones 2001 in in the eastern of Africa, also tropical countries, Rocha et al. 2001. In Brazil, also tropical countries, and Sequeira and also Rocha also in Brazil 2007. So the number of individuals higher in this study than those studies. The diversity index is higher than Fornes 2001 and Rocha et al. 2001, but the density significantly lower than Rocha et al. 2001 and Fornes 2001. It is maybe due to our area is a tourism object where some people frequently visit the area for spending the holidays with a large number of people, so it might be can disturb the habitat. This is what I said, that microalgae contain 5 species. This is the richest family, and Bijuran and Icobiose contain 5 individuals, which place this species to the highest number of species. And the species accumulation curve, so that after the plot 20, the new species is absent. So the curve is flat, hover after 20 plot. So our recommendation is smaller plot, 16 meter is preferred for small leaf-litter frog survey. And then researchers can allocate 2 hours per night by 2% to complete 15 plots. So it will minimize the cost for accommodation and consumption, and also it will save so much energy of the people in the survey. Thank you very much. Okay, that's the final video recorded for the presentation. Now we will turn into the discussion session. In the discussion session, we open the question for any presenters from you all. If you have any questions, please write it in the chat box or just ask directly to the presenter. And we already have one question coming from Ms. Afifatul Ahyar. The questions for Ms. Adiba is about DNA extraction method that you use to get the environmental DNA from water samples. What DNA extraction method you use for that? Ms. Adiba, can you hear my voice? Oh, yeah, it looks like Ms. Adiba is out from this Zoom meeting. So unfortunately, Ms. Adiba could not answer the question, but we will forward the question to Ms. Adiba so that she can answer later if she still cannot join this Zoom meeting. And we still open the questions from anyone to any presenters. Okay,